Hi everyone, welcome to my Abhyas. We are here with the new chapter, chapter number 10, that is Microbes in Human Welfare. This is a chapter where we're going to talk about the microorganisms which are actually invisible. We can't see the microorganisms with unaided eyes. We need to have a microscope to see that. Now here we are going to talk about the beneficial role of the microbe because it is human welfare. We are not going to talk about the harmful effect because already there is a chapter on that where we would have discussed about like what are the diseases the microorganisms causes and how they are going to harm our body. So we're not talking about the pathogens here. We are going to talk about the microbes which is actually helping us. Okay, let us see what exactly are microbes or what do you mean by microorganisms. Microorganisms are basically the very tiny invisible organism which are present on the earth's surface, and actually they are present everywhere. We say them microorganisms can exist anywhere, even if where the human cannot go or there is no other organisms will be no other organisms are alive. There also microorganisms can be living happily with all the having all the growth, reproduction, and so on. So the microorganisms are basically the tiny organisms which is a very small particles which cannot be seen, and we need microscope to see this. It was understood that microorganisms are the first organisms or first primitive organisms, what we can say them. Okay, they are the primitive organisms on the earth's surface. The life on the earth started with the microbes. When the earth was getting created, at that time there were a lot of reactions were going on. It is believed that there were years together of storm, there were years together of rain and volcanic eruptions and so on. During these type when the lightning and all were going on, when the earth is getting its shape, there were some big molecules started getting created, and on that basis the first big biomolecule which was created is supposed to be RNA, that is ribonucleic acid. This RNA and then later followed by that the DNA, that is deoxyribonucleic acid. So these RNAs and DNAs got a capacity to self-reproduce. That means they started doing replication of themselves, and that is when the life on the earth started. But still the scientists are clueless that how the RNA or the big biomolecule got the capacity to reproduce of its own, or how does it started dividing, how does it started replicating. That idea is not clear yet. But it is believed that from that single strand RNA or from that single strand DNA only, the life on the earth has started, and from there the whole world, that is what we see, the living organisms present on the earth is created. So from there, like you can understand that when nothing was there on the earth, there was complete empty earth surface without any organisms. At that time microorganisms were there, and microorganisms were actually started growing over there, and then slowly they started dividing. Followed by that there are a lot of divisions and a lot of changes has happened. What we see right now on the world is though there are variations in the atmosphere, there are a lot of organisms what we are seeing, but the first organisms are microorganisms. Where do we find them? They are present everywhere. Microorganisms are present everywhere. What they eat, what is the nutrients? They can eat anything. We say that microorganisms are omnipotent and omnipresent. They are present everywhere, and they literally can digest or can break down any substance present on the earth except the plastics. That is where we are still working. The scientists are still working that can microorganisms also degrade plastic. If that those microorganisms are discovered, then okay, very good. We are actually having a lot of pollution control we can do. But anyway, the microorganisms are not yet able to break down the long chain which is present on the plastic molecule. Other than that, almost every single thing on the earth is almost degraded by the microorganisms. There was an experiment which was done on that. It was proven that even microorganisms can break down the cyanide, which supposed to be the very poisonous for any living organism. Microorganisms can grow and reproduce and extend its colonies even on a cyanide medium also. So you can imagine that how big their world is, how many different types of microorganisms must be there, and there is a possibility that we don't know that still how many organisms yet to be discovered. There are so many organisms which has to be understood, studied, and maybe their beneficial roles we need to know about it. So that is what is basically what we call it as microorganisms. Where they're present everywhere. We can find them. If you take a handful of soil, you may find 1 million of microorganisms present on that. They're present on the thermal vents also. The thermal vents, which is like geysers and all, where the hot water flows there. There also microorganisms are present. Microorganisms are present even in a very snowy region also. That means in a very cold temperature also there are microorganisms. Even on the acidic environment also there are some microorganisms. Those are acidophiles. They can grow. That means that they can be present everywhere, anywhere and everywhere. And on our body, there are millions of microorganisms which is staying inside our body, outside our body, on our skin level, in our mouth. There are so many microorganisms which is there. So one thing comes over here when we talk about microorganisms, that they must be very, very dangerous. They must be very, very harmful. It is not like that always. Microorganisms are also helping us to do so many life activities. Actually, to be very proper, very frank with you, that without microorganisms, the life would not have been possible. Did you understand what I'm telling? It means that there is so much of help we take from these microorganisms that if we remove the beneficial role of the microorganisms from our life, life will stop at that moment only. Like that, they are important. Yes, there are pathogens, but at the same time, there are hundreds and millions of microorganisms which are actually beneficial. So we are going to talk about the good part now on this chapter. That is how they are helping human welfare. What are the help we are taking from the microorganisms? Okay. So stating that, let us first know how many different type of microorganisms which are possible. Microorganisms can be classified in terms of protozoa. Then there may be bacteria. A large kingdom of bacteria is large number of bacteria are present. Okay. Then there are protozoa, bacteria, fungi, funguses. They also comes under. Some of the funguses are visible. Those we can see through our naked eyes also, but they are considered as microorganisms. So protozoa, bacteria, fungi, and very importantly the viruses. Virus is such an organism which is actually having a half alive. That means they can only work, or they can only be alive when they are present outside. They can be only alive when they are inside the body of an host. Outside the body of an host, they act like a dead, or they act like a non-living thing. Okay, so that is how we can classify microorganisms as protozoa, bacteria, fungi, viruses, and these are having a lot of applications on our life. So let us see what exactly these microbes are doing to do the human welfare, how we are taking the benefit from these microbes. When we talk about benefit, why not to start with food, isn't it? Food is always good to talk about. So when we talk about food, does microorganisms help us to give any food, or do we take any help from the microorganisms to make our food, prepare our food, bake our food, cook our food? Yes, we do. Just think for some time. Can you tell me what exact help we are taking from the microorganisms? What comes in your mind when I say microorganisms and food? Okay, they spoil the food. That is a bad part. I'm not talking about bad part now. Just think that in this chapter, we're going to talk about only the good behavior of the microbes, not the bad behavior. They are now a part of our study where we are talking about only good about microbes. So let us see microbes in food industry or food world, how we are going to check the importance of microbes. So when we say that, the first example, let me give an example, then you will understand. The curd. Okay, what is curd? Curd is acted upon by the microorganism. That microorganism is lactic acid bacteria. This lactic acid bacteria will be working on the curd and working on the milk, and then coagulate the milk to form the curd. So, this is one of the very old process of coagulating the milk and making curd. Okay, so let us see how exactly it has been done, what exactly the benefit we are going to get from the microbes. So, curd is started, the fermentation of milk or the production of curd started a long ago, when it's what we can say, it's a by chance discovery by chance. So there was a starter culture which need to be added. At home, when you would have seen your mom or anybody preparing the curd at home, when we do make homemade curds, what they do, they mix 1 spoonful of curd to the milk, right? 1 spoonful of curd to the milk what is existing. So that is actually acting as that 1 spoonful of curd which is getting mixed with the milk is acting as a starter culture. What is the meaning of starter culture? That particular spoonful of curd will have a lot of bacteria which will help to coagulate the remaining milk present where we are mixing it. So this starter culture is basically containing the bacteria called as lactobacillus. Okay, lactobacillus is the bacteria, or it is also called as lactic acid bacteria. It is shortened as LAB, lactic acid bacteria. Commonly called as lactic acid bacteria. Generally, what it is done in an industrial process, milk is heated to 80 to 90 degree Fahrenheit, 90 degree Celsius. Milk is heated up to 80 to 90 degree Celsius, and then it is cooled to 45 degree. So this is the heated one, and then it is cooled to 45 degree Celsius. Okay, then it is cooled to 45 degrees Celsius, and the starter culture is added, and on this milk the starter culture is added. What happens now? It has to be this starter culture, as it is containing lactic acid bacteria, they start growing over there, coagulating the milk and producing the curd after some time. You have to give some time for the bacteria to grow, maybe 6 to 8 hours, so that the curd settles down and we get a yummy tasty curd. But what exactly the benefit of the curd? Some people cannot digest milk. The milk casein cannot be digested in the stomach of those person. So in that case, doctor and people will suggest that to have curd. In the curd, the the quality of the milk fortifies. It increases with vitamin B12. Okay. And in the stomach, we have lactic acid bacteria, which is already present, and they are helping these, curd to be checking all the diseases which can come from the microbes, as well as this lactic acid bacteria is helping us to digest the whole process. So that is how the curd is one of the very important product in our daily life. Almost every day we consume curd, and here we are using the microorganisms. Without the microorganisms, this lactic acid bacteria, the production of the curd would have not been possible. Okay, let us say one more thing, the second one. That is our favorite breakfast for so many people, that is dosa and idli. Dosa and idli puffs up. You would have seen that when we keep it, the batter of dosa and idli at home for a night, overnight, what happens is the dosa and idli batter puffs up. It ferments. When there is a fermentation going on, so there is one very important thing what you should know over here, that is called as fermentation. When the fermentation is going on, the batter puffs up to leave the carbon dioxide out of it, which gives the fluffy or soft idlis and crispy dosas. With that, it makes a small tiny holes because of the carbon dioxide escaping from this, and it is generally done with the rice. So rice gets fermented because of the presence of the natural bacteria which is present in the atmosphere, that start fermenting the dosa and idli batter, and we get a tasty food out of it. Okay, so mouths are watery, right? Mine too. Okay, so what exactly is happening in fermentation? Let me tell what is fermentation. In case of fermentation, the carbon dioxide will be released from the mixture. So if we have a batter which is mixed with rice and the gram dal, or what we can say the urad dal, black gram or urad dal, whatever is used over here, gram, we'll say black gram, or you can use urad dal. Okay, so when it is mixed together, grinded together and left for a longer period of time, the carbon dioxide will be released from this atmosphere, from the particular mixture, and that makes the batter fluffy and soft, and from there we get a nice idlis and dosas at home as well as anywhere wherever we want to eat. So that is one of the very important one on the food industry. When we say about when it comes to food, microbes cannot be ignored. We cannot ignore the role of microbes any time for the food, when we say the food is getting prepared, or there are different types of tasty food in different state, different place, the help we take from the microbe is you can't count it, that many help we are going to actually taking from the microbes. So then our next food product, what you're going to study about, one of the very important one used everywhere over the whole world, is the next very important food product in our life is bread. Okay, bread is one of the very bread is one of the important food product for all over the world. Wherever you go, you will find the different types of different varieties of breads, and every time whenever you see a bread, you think that there is a microorganisms which is used to make that bread. What exactly happens, you would have seen that bread is puffy and very soft, and there will be a lot of holes present on the bread. The dough which is used to make the bread is mixed with a microorganism called as yeast. This yeast actually releases a lot of carbon dioxide and ferments the particular dough. While fermenting, it also releases a little bit of acid present on that, little bit of different type of organic acids and alcohol are produced when the fermentation is going on. So that gives the taste a little more better than a normal one. Okay, so what exactly happens when we mix the yeast over there, that the scientific name of yeast is Saccharomyces cerevisiae. Saccharomyces cerevisiae. B-E-S-I-A-E. Saccharomyces cerevisiae is the scientific name of the yeast. So what it is done, yeast is mixed with the dough, and as it is kept for some time for the fermentation process, the volume of the dough increases. This increase of the volume by the adding of yeast is called as leavening. Okay, what is the meaning of leavening? Leavening is the process which makes the dough lighter, and it releases the carbon dioxide. Because of that, it becomes fluffy and bread becomes easily digestible because of the leavening process. Yeast also produces, as I told you, some of the alcohols and some organic acids. These alcohol and organic acids will give the taste, it enhances the taste of the bread. While the baking process is going on, because it has to go in the oven for the baking process, when the bread is getting baked, at that time this alcohol and organic acids evaporates, and the yeast microorganism yeast is obviously killed due to the heat process, and at the end we get a soft, puffy and very tasty bread. So that is what exactly is helping in this process. The whole process is only dependent on this Saccharomyces cerevisiae. Did you understand that? There is one of the very famous traditional drink which is used in different part of southern India, and that is one of the important fermented product which is used in a local places, that is called as Dodi. Dodi is a traditional drink generally made up of the by fermenting the sap of palm tree. Sap of palm. It is a very old process of making this fermented drink, which is again used. The fermentation, whole process of fermentation goes by the microorganisms. The microorganism ferments and gives it a different taste, and is very, very famous in the southern part of India. One more very important food item, which is my mouth is watery when I'm talking about this. When we say that there is one more important food product which is again used worldwide, that is cheese, right? Most of us have tasted cheese and know what exactly cheese is. Cheese is one of the oldest food item which was produced by using microbes. There are different varieties of cheese available all over the world, and that they vary depending on their texture, flavor, color and taste and so on. So there are a lot of varieties of cheese which is available everywhere, but that depends on what type of bacteria or fungus we are using to ferment the cheese. So what exactly making the cheese different? The cheese is different because the funguses and the bacteria, whatever we are using, is different from one another. And one of the very popular cheese which is actually used everywhere, that is called as Swiss cheese. Okay, Swiss cheese is is produced by the largest production of carbon dioxide. A large amount of carbon dioxide is produced when the fermentation is going on, and this cheese is prepared by the microbes Popinibacterium shermani. You should remember some of the name of these bacteria and the microorganisms which is required for your exam. Okay, so Propinibacterium shermani, Sharmani, Sharmani. This is the name of the microbes which is used to produce Swiss cheese. There is one more very important cheese which is again a very famous cheese, that is called as Roquefort cheese. Roquefort cheese. And to produce this Roquefort cheese, the microorganisms used is Penicillium roqueforti. Penicillium is a fungus. So this fungus is used over here to make the cheese. Penicillium roqueforti. By the name only we can say that, then you can easily remember the name of this, because how can you remember? The name of the cheese and the name of the fungus is same. Penicillium roqueforti is the fungus which is used to produce Roquefort cheese. Other than that, there are soya cheese, then there are fermented cabbage cheese, there are different type of pickles and sauce, and there are fermented cow's milk products. There are a lot of items, lot of varieties of things which we can prepare and which we use as a food in our daily life, in different industrial processes. It is used everywhere worldwide. So, when we talk about how many type of food items we are telling about the microbes, then you will forget the numbering. Actually, there are so many items where we actually take the help of microbes. So what basically microbes are doing? Microbes are very good in fermenting the substances. What is the meaning of fermentation? The microbes will be using the particular medium where it is grown, and it will start releasing carbon dioxide gas and ethanol. Sometimes some organic acids will be produced, sometimes little bit of alcohol will be produced. So while doing this process, when the microorganisms are growing, they are basically releasing carbon dioxide, and when they release carbon dioxide, that is one of the very important thing when we say fermentation. That makes the batter fluffy or the dough soft, and that makes the drink more tastier. And also, when we say the cheese production, there are a lot, there is a whole book can be written on how many different type of cheese are there and how many different type of varieties of microorganisms can be used in different ways to make cheese. Okay, so that is about in the food industry. And there is one more very important industry which helps a lot of economic growth in the whole world. That is the alcohol industry. Microorganisms is one of the integrated part of this alcohol industry. When we say that, how does the microorganisms are helping in this process? Microorganisms are basically, helping the whole batch of the fermentation going on at a time, and at the end of the fermentation process we are going to get different type of alcohols, and that is on that a lot of economy of that particular country depends on. Okay, so let us see that how the microorganisms are used in the alcohol production and in the fermenters. So basically in this chapter what we are talking about how microbes are used in the beneficial role as a benefit. What are the benefits we are going to take from the microbes? Previous chapter we have studied about the pathogenic role of the microbes. This chapter what we are talking about, we are seeing the beneficial role of microbes. So on that food industry we discussed. Now we will talk about alcohol industry, how the microbes are helping. Also you have antibiotics production, how the microbes helping in the production of antibiotics and vaccines. And at the last we have the waste disposal techniques and the biogas plant. There also microorganisms are used. So now we are going to talk about alcohol production and how a microorganism is helping in this process. When, as I told you before also, during the fermentation process, a microorganism is basically using a particular medium, and at the end of it, it is producing some ethanol and some acids, alcohol and acid. collecting those acids and purifying them in a different level, different concentration, we get different type of alcoholic beverages. There are a lot, like whiskey, rum, beer, and so on. There are so many. Okay, so there are so many alcoholic drinks which can be prepared by the use of the microbes. In all the production of all the processes of the production, every time in the fermenter, the microorganisms are used. Okay, so for the large scale production of the alcohol, a large culture, a lot of culture of the microbes is used for the fermenter, and then in the fermenter, the fermentation process goes on. So what is the basic requirement over here? There are big fermenters. In the fermenters, what is the basic microorganism they use? They use yeast or Saccharomyces cerevisiae. It is one of the famous or very common and popular microorganisms for the production of alcohol. Okay, so it is the Saccharomyces cerevisiae. Saccharomyces cerevisiae is the scientific name of yeast which is used in the fermenters as a starter culture. This is, as you already learned, that Saccharomyces cerevisiae is also used for the bread production as well. Okay, so what is the basically happening over here? What is the reaction? That glucose is getting fermented to form ethanol, that is C2H5OH, with a carbon dioxide gas produced out of it. So this process is called, yeast is used, and where the reaction is going on? In a fermenter. Okay, fermenters are large big tanks where the glucose mixture of different types will be used, and on that yeast will be added, and at the end of it, depending on the the distillation process, because alcohol, whatever is produced as a raw product, cannot be used as a beverage for drinking. There should be a lot of distillation process should go on to remove the microorganisms from there, to remove all the toxic substances, and depending on how much purification has been done, the different types of brands of alcohols which is available in the market. Okay, so depending on if it is done with a good distillation, or there is not the purification process is good or not, depending on that, the varieties of alcohol is available. So after the production of the ethanol over there, it undergoes a distillation technique. What is distillation? Distillation is a process by which the evaporation will be going on. During the, you have learned distillation in chemistry also, I believe. So in that, what happens, that there will be evaporation and the vapor will be collected and cooled down. So whatever is actually needed can be separated. It's a physical process of separation, can be separated from the whole mixture. So on that note, we have so many type of alcohols which is produced. Wine and beer are produced with distillation. Okay, beer and wine is produced with a lot of distillation. There are a lot of distillation which goes on when the production of wine and beer is going on. Whereas whiskey and brandy is produced without distillation. Whiskey and brandy is produced without distillation. In this process, they do not use the distillation, but they use some other purification method for making the drink tastier and healthier as well. Like, there should not be any toxic substances, I mean to say. Okay, so there are volatile components which is boiled to a particular boiling mixture, and on that the separation of alcohol is going on. So beverages and antibiotics are the two big industries where exactly the microbes are used, and a lot of studies and research goes on this sector as well. Okay, so the next point, what it is, that is called as antibiotics. So as we spoke about the different type of food items in the good role of the microorganisms, that means good role of the production of food products. In that we have the alcohol production, we have different type of food varieties which is produced. There is one of the biggest industry which is dependent on microbes is the antibiotic industry, which comes with the medical science and medical industries as well. What happens in those antibiotic production is microorganisms, as they're very, very small, they can be grown in a small place. Do you agree that? So there are experiments which can be done very easily with the microbes. You can just, grow the microbes in a culture medium, in a petri dish, and that can happen in a short span of time. The result which we get at the end of the reaction is quick. We need not to wait for a long time to see that if the reaction is happening or not, if the microbes are actually growing or not, if they are reacting for that mixture or not. Do you understand what I'm telling? So because it is easy and convenient to use a microbes, that is why in the antibiotic production industries use a lot of bacteria and sometimes protozoan and viruses as well for the production of antibiotics. What is an antibiotics? Anti means against, body means life. Okay. So what are antibiotics? Here the term is made up of two words. Anti means against, bio means life. It means against life. It doesn't mean against life of the host, but it's against life of the pathogen. Okay. So antibiotics is used for different purposes, for treating diseases and so on. So let us see. Antibiotic was first produced in the year 1929. Can you recall? By Alexander Fleming. Okay, by Alexander Fleming, who was working on a Staphylococcus bacteria, and he found out that one of the mold which is growing on the Staphylococcus medium, at that place the microbes growth is less, or the microbes are not growing at that particular region. So then he started checking that mold, what exactly it is, and while finding out those researching on, he was actually researching on Staphylococcus, but he discovered Penicillium, the fungus. Okay, Penicillium notatum. This is one of the first antibiotic which was discovered by Alexander Fleming. Penicillium notatum. This is the first antibiotic which is produced, and how much effective it is, that was out of discovery Alexander Fleming could not do. But later stages, when the World War II was going on, at that time this Penicillium notatum was used to treat the soldier in a large amount. There are a lot of life which was saved because of this Penicillium notatum. Okay, so antibiotics are produced both by the fungi and the bacteria, and 50% of the whole antibiotic can be produced by Streptomyces alone. Streptomyces species, the bacteria, not the species, the genus Streptomyces of this type of bacteria, Streptomyces, this can approximately prepare 50% of the whole antibiotics. Okay, so what exactly the antibiotic is doing? Let us find out some of the name of the antibiotic. Penicillin. Penicillin is produced by the fungus Penicillium notatum. Okay, let me clear this part because I may need this part as well. I'll give you some examples. This may be a common one for you. This may be required for your exam. Sometime it is asked in the NEET CET questions as well. Then let us see erythromycin. It is one of the very popular antibiotic. Erythromycin. Erythromycin is produced by Streptomyces erythrus. Streptomyces erythrans. Erythrans. Streptomyces erythrans. Then we have one more, that is neomycin. There are a lot actually. I can't give all the names, and you also can't remember all the name of the antibiotic produced and the name of the bacteria. But some of the common one, let us find it out. Neomycin is produced by again Streptomyces. I'll write ST for the Streptomyces species, genus, and it is Streptomyces freddi. Okay, Streptomyces freddi. Chloramphenicol, let us write one more. Chloramphenicol. CHLO, chloram. This is also one of the very important antibiotic used. They are broad spectrum. They can kill a lot of bacteria by giving one particular antibiotic can kill so many bacteria. Okay, so chloramphenicol, it is Streptomyces venezuelae. is the name of the place where it was discovered, Benny's Valley. Okay, so like this you can have several examples of antibiotics or the bacteria which is used to produce these antibiotics. The examples will make our board completely filled. I have given some of the antibiotics. There are some antibiotics which is specific for some specific diseases. For example, whooping cough. It is one of the life-threatening disease, which is azithromycin is the antibiotic for that. We have the antibiotic for leprosy, where it is dapsone and clofenzine is used for the treating leprosy. Then plague is used by streptomycin tetracycline, where the treatment of plague the antibiotic uses streptomycin and tetracycline. Diphtheria is treated by penicillin and erythromycin. So like this, if you list out the diseases which can be cured by the antibiotic, there are a lot. And in the medical industries, in the pharmaceutical industry and medical industry, the usage of the microorganisms for the production of antibiotic is very large. We are very much dependent on the microorganisms for the production of antibiotic. So as the same time, they understand two things over here. One, bacteria is causing a disease, that is they are called as pathogens. At the same time, to kill that bacteria, we are using one of the microbes to protect us. So do you see that there is a communication between both disease causing organisms and disease curing substance? Both of these are a microbial product. As they are causing a disease, at the same time they are also helping us to get rid of it, or maybe getting cure of it. Okay, so we'll stop our class over here today. We'll be coming up with the biochemical help, how their different type of enzymes are produced, and we also have to discuss one of the very important and commonly asked question on biogas. So on that we'll be coming up with the next video. So till then, you stay healthy, stay happy, appreciate the role of microbes, which is helping us in keeping us healthy as well. And thank you for choosing my Abhyas. Take care.